Okay, by my reckoning that's 11:30 start time. Thanks very much for everyone coming along to this extraordinary seminar. Late notice from Professor Mark Pallen, who's had a pretty long career in medical microbiology research. He's trained as both a medic and then also doing a PhD and medical work in London, at London School, London Hospital, and Barts. Then a PhD at Imperial College, and then went to Northern Ireland to Queen's College, Belfast.
Queen's University.
Queen's University.
Yeah.
Get some of this right.
Yeah.
Then on to Birmingham, and then currently at University of Warwick. There's also you might want to check out some of his work on the internet. If you want to see more after this, there's lots of talks and things by Mark on YouTube. There's also a book which I noticed on Amazon is available from 1p, which I think.
Well, that's the way Amazon is, yeah.
Which I think either means that you've saturated the market, but I'm sure it's not the alternative because there's 13 reviews and they're all 5 stars. So
the reason it's 1p is because the Vice Chancellor in Birmingham, in his wisdom, decided to give the book out to all 6,000 first-year undergraduates one year, and obviously a large number of those 6,000 said, "Oh, I could just sell this."
Trying to make a tidy profit.
Yeah.
So that's a book on evolution, and I saw many of those topics going to come up today. So thanks very much for coming on, and I think
great. Okay, well, thank you very much for the invitation. So I'm going to talk about some things I haven't spoken about for a while, and put them in a rather slightly strange format. And you kind of hinted that I'm getting on a bit. I'm a certain age. Some people in the audience may see where I'm coming from. We'll see in a minute. But basically, I'm going to draw on 3 muses: the legacy of Charles Darwin, Theodor Escherich, and Fred Sanger. And you can see that they kind of overlap-ish. Darwin and Escherich did. There was a little bit of a gap between the death of Escherich and the birth of Fred Sanger. Basically, we're not far off having a continuum that stretches back into the early 19th century. But is there anyone in the audience who's getting where I'm coming from in using this particular style of presenting these 3 individuals? Yeah, it shows a certain certain vintage. All right, I can't see anyone in the audience who's knows about this, but there was a book came out back in the late 70s called Gödel, Escher and Bach. A copy of it here. It's a 20 years anniversary edition, and it's a remarkable book. It's a very interesting play on the legacy of Gödel, Escher and Bach and the ways that those things interact with each other, and it's also about what it is to have a mind and how can a mind emerge from kind of mindless matter. But what the key thing is, it was very playful. There's a lot of kind of Lewis Carroll type stuff in there as well. I'm getting the feeling that nobody in the audience has ever heard of this before, so probably none of you will also... Someone at the back there did put their hand up, did they? Yeah, okay. So why does actually having any interest in this book put me in bad company? The pictures are good. Yeah, we show but actually liking this book as a microbiologist puts me in extremely dodgy company. And the reason is that the guy who actually put anthrax into the American postal system had an obsession with this book, and he used certain abbreviations. He basically, in his notes, saying that he'd sent with the powder. He'd underline various characters to make up codons and various word games, the kind that were played in this book. And when the FBI came to investigate him, he actually threw the book out, and they found the book in his rubbish in his house. So it's kind of interesting. So maybe I piqued your interest. Nothing to do really with microbiology. There's quite a bit about DNA in there, but it's from the late 70s, so don't expect too much molecular biology. Right, so what I'm going to do is cover Darwin and evolution, Escherich and E. coli, Sanger and sequencing, the evolution of technology, and then a metagenomic diversion in the style of Escher, Gödel, Escher and Bach, and then talk about future direction. So, as you can see, I've decided to try and cover quite a lot of areas, and I might go through them a bit too fast. So what I've done is I've prepared an online guide. So those of you who've got the right app on your phone can just scan that in immediately, or you can just type that in, and you'll get a guide that will actually point you in the direction of, as you pointed out, there are plenty of other talks on YouTube that go on to some of these points. And if you want to access the original papers, there are links to them all there as well on the online guide. So let's start with Darwin. So when we think about Darwin, we think of this old guy here with this big bushy beard, the one they put on the £20 note, make it harder to forge. And Darwin gave us a huge grab bag of concepts and ideas. And if you read Darwin's work, The Origin of Species, for example, it's quite striking that although it obviously is written by someone in the middle of the 19th century, many of the ideas are still very fresh and current today. And it is just a warble of some of the concepts that Darwin grappled with. And many of them, even in this age of genomics, are still very fresh and meaningful. And that's quite remarkable, given that Darwin didn't even know about genes, let alone genomes. It's still a remarkable legacy there. So there's The Origin of Species. Is all of Darwin's works on my bookshelf. You can see that I haven't broken the spine on most of them. I'd be a fraud if I pretended I'd read all of them. I've read The Origin of Species, and I wrote this book. So that gives me some, just a little bit of credibility to talk about Darwin. In fact, in my inaugural a couple of years ago, I pointed out I'm not obsessed with Darwin. I did quote him at my wedding. My daughter is called Emma and my son is called Charles, the first two children. I'm on first name terms with two of Darwin's descendants. And as I pointed out earlier, we did get Randall Keynes to open our bioinformatics centre recently, who's Darwin's great-great-grandson. But anyway, let's move on. One of the things when I was preparing for that book was I got the chance to sort of look at Darwin in a bit more detail than usual and looked at his notebooks. And the interesting thing was that here you've got a different view of Darwin. So this idea of Darwin as an icon, as a bushy beard and that. The young Darwin actually was just as much a kind of degenerate young man as any student is today. I mean, he wasn't quite, the sexual revolution hadn't happened, so he wasn't quite as straightforward as things might be now. But in his notebooks, you find this interest in sex. He talks about a man who wanted to have sex with a turkey. It talks about, why their nipples on men and women, what's the clitoris for, all those kind of things go in these notebooks. So this kind of riotous mind, and as a student, he was a complete waster. He drank far too much, but the notebooks are interesting because they're kind of like tools to think with, if you like. It's one of the few cases where we can actually see the evolution of thinking in someone's mind. So there's this set of notebooks here. This is one of the first sketches of an evolutionary tree that he wrote. So some of you have probably seen the more iconic I Think tree, but this was an earlier one where he tried to point out that basically there's this branching evolution and the dotted lines of the extinct lineages, and then and he had all these thoughts, basically in his early 30s. He was sketching this away. And one of the things that I find is one of the most remarkable lines that as he was thinking about this, he came up with this here, that if we elect conjecture wild animals, our fellow brethren in pain, disease, death and suffering and famine, are slaves in the most laborious work and companions in our amusement. They may partake from one origin, our origin in one common ancestor, we may be all netted together. That's effectively the first statement of animal rights, if you like, and of the vegetarian movement and that kind of thing. But it's also the first statement of the One Health agenda, which is very much in people's minds at the moment when thinking about human health and animal health and looking at the same continuities between them, and that's all down to Darwin. But anyway, what did Darwin actually say? Well, this guy Ernst Mayr, another one of these remarkable individuals who straddled eras, if you like. Huxley was still alive when Ernst Mayr was born, and he only died a few years ago. He basically, in a paper, said, well, what did Darwin actually give us? He gave us a notion of branching evolution and common descent. natural selection, gradualism in evolution. But interestingly, Darwin put a little spin to say that actually the biological sciences, evolutionary biology, has a historical angle. And that in evolution, we're reconstructing events that have already taken place, which is a bit different from chemistry and most of physics, at least. But the other key point was that it feeds on variation and randomness. So in evolution, we look at the innumerable variations between individuals in the same species as something interesting, because that's what evolution builds on. A physicist will take one electron in the universe and see it as a representation of all electrons, are all effectively the same, whereas all that messiness is actually put into the foreground by Darwin. And the key point was it banishes this typological thinking that you can sort of classify things very strictly into a particular box and keep it there, when in fact in evolution things don't quite behave that way. Moving on to Escherichia coli and Escherich. So this bacterium was discovered by this guy, a German-Austrian physician, Theodor Escherich, in 1885. He first isolated it from babies' nappies, one of those things that grew very well. And the name Escherichia coli was first put forward in 1919. It wasn't actually formally adopted until I think the late 50s by taxonomists, but it was named after him. And there's a little side allusion here to Escherich. To Escher from Escherich. This is one of Escher's iconic pictures, but instead of him holding a globe with a reflection, he's holding a bit of E. coli in close detail. So one of the interesting things, Darwin made a lot about domestication. First chapter, The Origin of Species, all about domestication, and he saw great truths in studying domestication. But if you think about what we've done to E. coli, we've effectively domesticated this organism. And one particular strain, K12, was isolated in 1922. Again, just should say feces, not faces, of a convalescent diphtheria patient at Stanford University. This particular strain was then kind of gained a new life. It was taken up by Beadle and Tatum, or Tatum in particular, and then Lederberg used it. And then all sorts of key discoveries were made using E. coli in the history of biology. So the first proof that variation, that mutation precedes selection, came from this Luria-Delbruck experiments using bacteriophages that actually predated on E. coli. Then Hershey and Chase showed us that DNA was the genetic substance by looking at the behaviour of bacteriophages on E. coli, and obviously the semi-conservative replication of DNA was established in E. coli. So E. coli started to be domesticated in the pursuit of knowledge, and this continued, the discovery of operons, genetic code, but then actually in the 1970s the use of E. coli genetic engineering meant that it really did become domesticated for use for human benefit in a clear and obvious way, and now we have millions of diabetics all relying on insulin made in E. coli. And since then, more recently, we've seen systems biology and synthetic biology both relying heavily on E. coli. And in fact, Carl Zimmer, an American journalist, wrote a nice book a few years ago called Microcosm, where he basically summarised the role that E. coli played, foundational role in the emergence of science. Now, during all this, some people got a bit carried away. I don't know whether it's French people always come up with these kind of gnomic phrases, but Jacques Monod came up with this phrase that's all that's true. We said it in French, but I'm not going to try and murder French language for you. All that's true of E. coli is also true of the elephant. So basically he was saying that what you discovered in E. coli, you could generalise it across the whole of biology. Of course, there was a second narrative, though, with E. coli, because E. coli is not just a commensal, it's not just a model organism, and in fact, way back in the early part of the 20th century, the 1900s, Dudgeon and Ross described E. coli as a cause of urinary tract infections. And then as the 20th century progressed, there were a whole number of other landmark studies that showed that E. coli was, well, the Nissel strain was actually isolated in 1917 and showed that this could prevent infections. All the other issues here are to do with how E. coli causes infections. So E. coli is a pathogen, and it's also a probiotic in addition to being a commensal and a model organism. In the late 1970s, Sanger invented DNA sequencing, so that's where Fred Sanger comes in. For those who are fans of Fred Sanger, I'm not going to say much more about the guy than that, but he invented sequencing. It is worth stressing, of course, I say this in an audience like this, we can say this, but it hasn't escaped my attention that the coolest forms of sequencing are always invented on this side of the Atlantic. So there was maximum Gilbert sequencing invented by some American guys, but Sanger became predominant, and if you've noticed recently, things like 454 and iron torrent invented over that side of the Atlantic, whereas Illumina sequencing has become predominant again. Anyway, the other point, if we're being slightly facetious there, is that Sanger actually sequenced Phi-X174 with his new sequencing method, and that's effectively the first microbial genome to be sequenced. So we all talk about microbial genomics as if it burst out onto the scene in the 1990s, but in fact Sanger was doing it back in the 1970s. And in fact he sequenced the mitochondrial genome as well. And I have written a paper in which I argue, according to evolutionary cladistic taxonomy, the mitochondrion is a bacterium, and therefore this is a bacterial genome. So Sanger sequenced the first bacterial genome. And forget about those Americans, Craig Venter and all that stuff. Late 1980s, this was a remarkable moment for me when I realised that basically you could study evolution in bacteria, and it was all down to this guy Carl Woese, who initially using various fingerprinting approaches, then using sequencing, established that you could come up with a phylogenetic tree. Not only did he make that breakthrough in technological terms, in conceptual terms, but he also discovered a whole new domain of life, the archaea. So he has to stand as one of the great guys in the history of our field. But of course, if you go back to Darwin, you remember that little sketch in Darwin's notebook? In fact, if we went back to the notebook and we used a special antidote to the invisible ink that Darwin used, you can see that Darwin did actually get there in his notebooks in the 1830s and recognised the three domains of life. So perhaps Woese wasn't as inventive as we thought. But anyway, this is a lot of preamble here, but we'll get down to some key points in a moment. One of the issues for me was that when I first went to Birmingham, I started coming up against people who were working on E. coli as a model organism. But I was quite troubled by their attitude, really, because their view of what it was as a model organism was like, K12, I don't care about its evolution, I don't care about its ecology, it's just there, we use it, it's our model. It's almost like God created this thing for them to go and do all their studies on promoters or gene regulation, or synthetic biologists to this day are still treating E. coli K12 as if God invented it. Systems biologists the same. But in fact we have this history of variation under domestication, to use Darwin's chapter heading. E. coli K12 was isolated in 1922, but then underwent a number of steps which actually deliberately ensured that it was nothing like the original wild-type organism. And it became progressively more mutated, it became rough, achieved all these other changes. And this is a problem because we've got this typological thinking, and about 10 years ago I wrote a paper where we argued that actually using these laboratory strains of E. coli, we are ripping E. coli out of its context. These are deceitful delinquents growing old disgracefully, was the view that we took. And it's a bit like if you took a fresh-faced E. coli like this and you leave it for a while in a kind of degenerate environment and it turns into that, and then you leave it a bit longer and it becomes an icon. And this again talks about the kind of things that go on in this book, that basically things become images of themselves rather than being a thing of themselves. And there was this concern that K12 was like that. Now in the late 1990s we had the genomics revolution come in, and we started to see in exquisite detail the inner workings of some of humankind's most fearful adversaries. Here are some of the genome sequences I was involved in. And the analogy to me at that time, to kind of quote Keats, it then felt like some watcher of the sky when a new planet swims into his ken. It was just shortly, just a few years before, that we started to get the first pictures of the solar system in detail, and it was very similar. We were seeing the most fearful adversaries, microbial adversaries of humans, things like the causative agent of the Black Death or the White Plague, tuberculosis, we're capturing them in silico on our computers and actually getting to grips with them for the first time. But what we found with E. coli was that actually it was behaving it was a slightly odd organism. So again, take another of Darwin's chapter headings, variation under nature. We kind of knew already before genomics that it was a highly versatile commensal pathogen and probiotic. But what we found when we started using genome sequencing, that mono was plain wrong. His elephant analogy was misleading because what was true of one E. coli strain wasn't even true of the next, let alone of an elephant. So this is those who like to play conference bingo, this is one of the pictures that you put up when you're talking about the evolution of E. coli and genomics of E. coli. This was the first uropathogenic E. coli genome sequence paper, and the figure here showing that from the first 3 E. coli genomes, only less than 40% of the genes in those genomes actually held in common between all 3 strains. And that each of these strains had an appreciable number of genes that were strain specific. So here, 20% of the genes in UPEC were strain specific. So this was a really kind of a strange thought because they're all part of one species, but there's so much variation. And as time went on, I set up a database called Colibase that then turned into a database called Xbase. And here's just an image we produced from that. The green bits are the bits that are held in common with K12, the supposed archetype of E. coli. But all the red bits are the bits that are variant and different. And you can see that all these other strains have got huge amounts of diversity, and hardly much more diversity in Salmonella, which is a completely separate genus, let alone separate species. So it's kind of like a TARDIS-like arrangement here. There's more on the inside of E. coli than there appears to be from the outside. It's just a really rather strange finding. So our first foray into this, was looking at enterotoxigenic E. coli. So enterotoxigenic E. coli is kind of E. coli, as its name suggests, produces enterotoxins, and these are on plasmids. And there was a lot of interest in, can we build a vaccine against ETEC? Can we build something on the chassis, not just on the toxins, but can we take core components of the genome and do something with them? What we found was when we did phylogenetic comparisons using an approach called MLST, which had just been introduced for E. coli at the time, we found that actually there would be multiple acquisitions of these toxin genes by ETEC strains, and the ETEC strains is not a particularly good picture, I'm afraid, but the ETEC strains were scattered throughout the whole phylogeny of E. coli. So the idea that we could define a type, an ecovar or pathovar of E. coli, and call it ETEC, and think we could find something essential about it apart from the toxins, that was basically thrown out the window. And in fact, when the ETEC type strain was sequenced, there was found a considerable overlap between the type strain and commensals in terms of their gene content. So basically the genomics very quickly made us realise that it was trying to capture the essence of that organism and make a vaccine against it was going to be a hard ask and quite difficult. But we also found another feature that would have been very familiar to Darwin, the idea of vestigiality. So if you look in the E. coli K12 genome, and here's a little cartoon showing some of the genes here, you get this funny pair of genes here, in the middle there, where the coding sequences are actually kind of like fused together. And if you look by homology, they look a bit like flagella genes. And there they were. And this was a time when people were seriously starting to try and overexpress every protein coding gene in E. coli, to knock out every gene, to find a function for everything in the genome. But it never occurred to anyone that these things could actually just be remnants of something else. And it was only when we sequenced the first interaggregative E. coli genome at 042 that it became clear what was happening here. And at that point in the genome of 042, there was this huge, great, over 40 genes present there in a cluster which encoded an entirely new second flagella system in E. coli that had never been glimpsed or thought of before, a so-called lateral flagella system. And to my knowledge, nobody has yet found an E. coli in which it's functional, but the genes are clearly there. and you can compare them to lateral flagella systems from other organisms, and they all looks to be present and correct. But what you found in the supposed type strain is just this little scar where this has all been deleted, and just the remnants of the two genes at either extreme are left there. Now that might seem as if, well, and this is the case in the vast majority of E. coli. They're like K12. They just have this little scar, this little remnant of this thing. Now you might argue, well, isn't that obvious? Isn't it obvious you're going to see these things? But at the time, I think we all got carried away with this typological thinking and started to think, well, that K12 is in some way the kind of ancestral state or the archetypal state. And it was a bit of a surprise to find these remnants. We found another one called ETT2, which is a type 3 secretion system locus that had been broken in E. coli K12 as well, and in other E. colis. And in fact, I around that time started getting interested in the evolution of type 3 secretion, a particular form of protein secretion found in bacteria, which gives rise to these complex organelles associated with the cell envelope projecting outwards that are involved in secretion of proteins. And they basically fall into two groups. One group of type 3 secretion systems build the bacterial flagellum, the chief organelle of motility, and then the other ones are involved in building a kind of molecular syringe that allows the bacterium to inject effector proteins into eukaryotic cells, into host cells. And the interesting thing about these systems is that they are complex, and they kind of, you have to deploy these multi-protein complexes in a very tight regimen in terms of time and space. In fact, some people, you might call them idiots or idiots, intelligent design advocates, have said, well, it's too complex to even have evolved. It's proof that Darwin was wrong and that God exists. And in fact, if you want to read my counter-argument to that, I did write a review where we argue that that wasn't the case. But what I was interested in was a particular kind of E. coli, working with Gary Frankel in Gordon Dugan's lab, called EPEC and EHEC, that produce a particular type 3 secretion system encoded by this locus here, the locus for enterocyte effacement, or the Li. And when we first looked at the Li, if you're thinking about the way in which you would design something, you'd make it modular. Here is a nice gene cluster here, and the way it's coloured here is it's coloured by GC content. And in bacteria, if you get deviations from the average GC content, that's usually a signature that this is horizontally acquired DNA that's come in from outside of that particular lineage. So you can see here, there's this nice green splodge here where the GC content is very low. It looks like a kind of discrete system, and this encodes all of the stuff needed to make the molecular syringe, but it also includes 7 other genes which are involved in making the effectors, the things that go through the syringe. So you think that makes sense. If you were designing it as a human, that's how you design it. You design it so it's modular. It's got the, I think Gary's made the point before, you can call it the hardware and the software all in the same place. But nature doesn't evolve like that. So I got involved in kickstarted a project where we looked for more effectors, because when we looked at things like Pseudomonas syringae at that time, there were, I think, about 60 effectors in Pseudomonas syringae. I thought, well, how can E. coli be so poor in comparison to Pseudomonas syringae? This is E. coli. This is the master organism. It must have more effectors. And so we started getting together over 300 different effectors from all the different organisms we could find, and using bioinformatics approaches, using multi-searches, and then looking in the genome, we came up with 62 candidate effectors that were scattered across the chromosome. And they were found in 25 loci. So I, again, slightly playfully on words, I called them exchangeable effector loci because it was Lee backwards eels. And I think Roy Chowdhury at the time was a fan of the bandit eels as well. We then also worked with some guys in Japan, Toru Tobe, who made a SEP-L mutant, which over-secretes the effectors. So normally the effectors going through these syringes are only made when the syringe engages with the host cell. But if you knock out this particular gatekeeper protein gene, then it just chucks them out. So they were then looking at the secreted proteomes, and they found that they could find 31 secreted effectors, new effectors that were found using this approach. And then we actually proved, this was working with Gadi Frankel, that you could actually do so-called translocation assays where you could fuse markers to these effectors and show that they are actually not just being secreted out of the cell, but they're actually out of the bacterial cell, but actually translocated into the host cell. And so we ended up with 39 effectors confirmed experimentally as a result of this survey. But the interesting thing, I mean, it was interesting to find that there was so much more complexity going on in E. coli than we imagined. But the interesting thing is when we look at the genomic situation of these effectors, they're scattered around the chromosome. So the leads over here, you've got these effector loci all over. So it's not behaving in any kind of modular way at all. It's a strange situation where you've got this effector repertoire being run from lots of different sites of the chromosome. But it gets more interesting than that, because actually it then became clear that the majority of these are actually phage encoded. And what you have is you have the genes encoding all the structural parts of the bacteriophage over here. This is GC content again. You can see they're all kind of on the reddish side of the spectrum. But then these are actually much more on the greenish side, on the low GC side of things. And you can see this, it's almost like a cliff. You go along in GC content through the phage, and then you get to the effectors, and it just goes to the other direction. And so we had 3 clues that these were effectors. We could see by their location in the phage, because always at one end. We could see by the fact that they have this deviation in GC content. But also many of them, or most of them, well, all of them because of the way we did it actually, had homology to other known effectors. So this was a strange finding, and it made it clear that evolution doesn't work in the way that you'd expect, and it's much more versatile than you would expect from your designing things. Now, how am I doing for time? About halfway through, so this is about right then. So that was done somewhere about here on the graph, and even at that stage, this is again a slide you have to put up if you're playing conference bingo so people can sign it off. Basically, sequencing after Sanger invented it was obeying Moore's law. It was keeping up with computers and getting cheaper and easier as time went on. But then something happened around the 2007-2008 mark, where it suddenly went to what we might call hyper Moore's law, and that was the advent of high throughput sequencing. So Sanger sequencing evolved. So the legacy of Sanger continues, because at least these early high throughput sequencing approaches were still sequencing by synthesis, which was Sanger's great breakthrough. But they put the foot on the accelerator greatly. We had 1,000 times faster, 1,000 times cheaper. And we get this very lively competition. And then it got even better, because we went from having fairly largish kind of instruments. From 2011 onwards, we got down to benchtop sequencing, where it was easy, quick and cheap enough to have an impact on real-world clinical problems. And around that same time, as benchtop sequencing was coming in 2011, this rather nasty outbreak, Waggers called it the sprout break, an outbreak of E. coli, sugar toxin-producing E. coli in Germany, that caused 5,000 cases, over 5,000 cases, and over 50 deaths. It was linked to sprouting seeds. Initially, the epidemiologist got it wrong and said it was Spanish cucumbers and caused tremendous damage to the Spanish cucumber industry, but it turned out it was fenugreek bean sprouts that were the cause. There was a high risk of hemolytic uremic syndrome, the nasty consequence of infection with this E. coli. And for some reason, females particularly at risk, and we don't quite know why. Maybe that real men don't eat salads. Maybe there's some other reason, I don't know. But if you look at where it was affected, it was mostly concentrated in northern Germany, around Hamburg, but there were cases scattered throughout Germany, and there were cases scattered throughout Europe as people who'd been in Germany came back to their host countries. So there were, I think, 5 isolates made in the UK. Now, I was contemplating, how can we get involved in this and actually use our own talents to move things forward. By this time I had a guy working for me, Nick Lohman, and I said, well, why don't we have two plans? We'll have a plan A and a plan B. Plan A is we'll work with these guys in Germany and China and see if we can get something out of that. And plan B was, I know this guy John Wayne, who's working at the HPA at the time. Maybe we could get a UK isolate off him, and we could do something interesting with that. So this was just cunningly trying to make some use of this opportunity. So plan A turned into something called open source genomics. So what actually happened was that in Germany they had got plenty of copies of the isolate, plenty of isolates from the outbreak. They sent DNA from one of them off to BGI, the Chinese Genomics Institute, and BGI had just taken receipt of one of these things here, an iron time. So they were still trialling it. And it was known that there was a race on to get the first genome sequence of the outbreak strain. So the Chinese got on and sequenced it directly. And because they wanted to get their marker down quickly, they just released the genome sequence out into the public domain. In fact, they released it under a Creative Commons 0 license, which basically said, here's the sequence, you can do what you like with it, you don't have to attribute anything, and so on. So then in my group, Nick Lohman had actually, he was at a bioinformatics conference on public health and bioinformatics, and he came across this and thought, this is interesting. So he sent out a tweet saying to all the other people at the conference and elsewhere, hey, look, this is interesting, why don't we analyse this? And what he did was he did his own assembly, first of all. And so he kind of added value to the sequence, then put it back into the public domain. And the reason he was able to do that was I just won an iron ton in the European-wide competition, so we were kind of gearing up to using it. The next day, a guy called Mike the Mad Biologist, that's the name he uses when he's blogging. His real name's Mike Feldgarten. But he said, well, this is not some kind of Andromeda strain that's come from outer space. It clearly belongs to an existing lineage of E. coli. By day 5, we actually had a strain-specific test. And by day 7, there were over 24 reports on an open source wiki describing the biology of the strain reconstructed from this genome sequence, and all this done by bioinformaticians working in their spare time for the most part. None of them were involved. Well, none of them professionally involved in public health in that regard. So we were very happy because we got all this, and we showed that social media, blogging and Twitter, could actually augment academic discourse, and we showed that there was a key role for benchtop sequencing. And I was given the task of actually drafting the final paper. We wrote a paper which got published in the New England Journal of Medicine describing all this. And the interesting things that came out of that was that basically it was, it's a wake-up call for people who think that infection's not really a problem anymore. Here was an infection that went into one of the most advanced societies on the planet and created this devastation. And it was not a new strain. It was something that had been seen before, moving around very quickly. In fact, David Studholm, I think when he was on this site here, was the one who showed that actually it was most closely related in terms of genome sequence to a strain from the Central African Republic. But it belonged to a lineage that has previously been described as an interaggregative lineage. So it was a sugar toxin producing E. coli that belonged to an interaggregative lineage. So again, this was another challenge to typological thinking that we should have abandoned perhaps, but we still keep going back to. It's clear that pathotypes can evolve and overlap, and that virulence factors can jump around. And one of the interesting things, people like myself and Gadi Frankel, whenever we used to write a grant proposal in around the turn of the millennium, we'd always say sugar toxin producing E. coli is a really, really serious problem. And it produces sugar toxin, all right, but it also needs this thing called the Li to cause disease, and that's why you must give us loads of money to work on the Li. And this came along, and it didn't use the Li. It was able to cause disease without the type 3 secretion system. And also we saw antibiotic resistance in this strain where there had been no prior use of antibiotics, so it's clear that this is an emerging problem. Plan B I'm not going to say much about. Basically, Plan B was working with this guy here. Now, you wouldn't buy a used car. Someone who frowns quite like that. Yeah. He's not in the audience, is he? So I can be as rude as I like. But he gave us, he had an isolate, and I said, well, what are we going to do? I said, if we go in and just sequence its genome and say, here's a sequence of an English isolate from the outbreak, it's going to be a bit underwhelming. Can we put a spin on this some way different? I said, well, you've just got a 454, haven't you? 454 gene. He said, yeah. I said, we've got an iron type. We could compare them. And he said, actually, he could probably get his hands on a MySeq as well, the early MySeq one. And so we managed to sequence the same genome on 3 different platforms. And Nick Lohman went away and did all the analysis of the information we received and came up with this paper, benchtop comparison, performance comparison on benchtop high sequencing platforms. And amazingly, we hit the literature just at the right time. We showed effectively that the Illumina platform was by far superior. It was much easier to use, and it was much more accurate. And we confirmed that the iron torrent had the same problems at home with polymers as the 454. And we've ended up with over 700 citations to this paper in less than 4 years, which is truly amazing. So John thanked me for helping uplift his ref submission last time round with this paper. So we got our paper in New England Journal in Plan A, as I say. And it was only after all that had come out, we then went over to Germany and we met the guys at the heart of the outbreak, this Martin Epfelbacher here, who was the head of the lab, and we had some champagne together. And we said, well, what are we going to do now? How are we going to show that we're just not a one-trick pony? You know, people will say, oh, you got lucky in the right place, right time, you know, you got a New England Journal paper, so what? So I said, yeah, okay, let's see what we can do. What's your unique selling point? And what's my unique selling point? I said, our unique selling point, myself and Nick, we know a lot about E. coli, we know a lot about genome, bioinformatics, that kind of stuff. What's yours? He said, I got a freezer full of shit from the outbreak. So I said, well, why don't we just sequence the shit and see what we can make of that? Sorry, I'm lapsing into vernacular, but we did kind of joke we could make a rap song, sequence the shit. Now, there was a context to this. Why would we want to do that? Well, if you look at the evolution of technology, in the way bacteriology works, even today, we're confronting 21st century problems but using 19th century techniques. So the gram stain was invented back then, just after Darwin died. Darwin was still alive when Koch actually showed you could isolate bacteria as colonies on solid media. So these two approaches of microscopy and culture have been around for well over 100 years. Couldn't we do something better in this modern genomic age? So what I suggested was, well, okay, we'll take your shit sample. Now, in the original paper, we took those fecal samples, we isolated the E. coli, and then we sequenced it. Could we not just get the same information directly by sequencing the stool samples directly? So we got our German collaborators to send us some samples, and the very first sample we looked at, we were very pleasingly encouraged. It was one of those things where I thought, well, if we got even just a couple of sequence reads that align with the sugar toxin gene, then we've kind of got an output. We've diagnosed sugar toxin producing E. coli. But what we ended up with was actually getting over 20-fold coverage of the E. coli outbreak strain. When we aligned the reads from the metagenome of the stool sample against the E. coli outbreak strain, it was there in this quite considerable abundance. Interesting, we get this kind of smile, which is the sort of thing you see when a bacterium is growing because the origin is overrepresented. So there's an upswing here, which is where the bacteriophage that actually encodes the sugar toxin is found in the genome. And so what we take that to mean is that's probably detecting DNA associated with the phage particles as well as those associated with the E. coli cells. Nick was able to actually pull out the sequences and work out what sequence type this strain belonged to as well. So that was all very, very interesting. And the thing was that we didn't really expect this to go so well, because the kind of textbook thing is that E. coli is a bit player in the gut. If you talk to gut people interested in the gut microbiota, they say normal people, it's the Firmicutes, and the Bacteroidetes are the major players, and E. coli is kind of, But what actually happens in disease is that clearly the E. coli blooms to large amounts and actually takes over that microbiota. Then we did this with various others, and this is just a quick look at some others. So we've got various, there's another E. coli, here we've got less coverage, but here we've got one where we've got over 500-fold coverage. In each case you can see we're seeing those bacteriophage probably associated genes there. What we also said to our German collaborators was, don't just send us stuff from patients you know who've got E. coli, the Escherich, but send us stuff and some samples from patients in the outbreak time window who had diarrhea, but you didn't know what they had, you didn't know they were part of the outbreak, but it turns out they weren't, and we'll see what we can find in those. And what we found was we could actually detect other organisms, so we could detect Clostridium difficile in one over here, Campylobacter jejuni here. Obviously not giving quite as tidy a plot as you get with E. coli, but nonetheless there. But interestingly, we got this thing called Campylobacter concisus come up in one of the samples that the Germans had said, oh, this is a patient who had C. diff. But we found a much higher number of samples from number of reads from Campylobacter concisus, which I'd not heard of, but it is described as an emerging foodborne pathogen. So basically what we'd shown is this approach of shotgun metagenomics could discover the things you weren't expecting. Wasn't just going to confirm your suspicions, but it could actually give you completely new information where you didn't expect it to be there. So we started to try and write this up for the Journal of the American Medical Association because they're having a special issue coming out on genomics in medicine. And we sent them a summary, and they spoke to their expert, their chosen expert, and my spies tell me, and confronting chosen expert face to face, I can confirm the chosen expert was David Relman, and David Relman said, this is cheating though, isn't it? Because they're kind of just aligning and mapping against what they know after the event. What they should do is put themselves in the shoes of the people facing that outbreak at the time it happened, who didn't know what the cause was. So they should be able to get that outbreak strain genome out of all that material without cheating. It's a bit like you can do a jigsaw puzzle much easier if you've got the lid and you can just align all your pieces against the lid. So try and do it from scratch. So Nick went away and did a couple of all-nighters. He's a very bright guy, Nick Lohman. And he said, right, he's got to start doing some simplifying assumptions. So he did this kind of blobogram or scatter plot, or whatever you want to call it, where he basically looked at visualizing the whole metagenome in these meta-metagenome, because he took 40 stool samples, and he assigned them by taxonomy to various taxa, and then so I don't know whether normally there's a key there, but it doesn't show you the key. But basically, the pink ones are the Enterobacteriaceae, but you can see that they're swamped out by all sorts of other taxa present, and the GC content smeared out there shows that there really a lot of taxonomic diversity in there. So then what he did was he said, well, let's actually make a simplifying assumption, say that if this is an outbreak, we should be able to detect the sequences associated with the causative organism of the outbreak in at least half of the samples in front of us. So let's chuck away everything that's not present in at least half of these samples. So when he did that, you can see he simplifies the taxonomic mess somewhat. You've got this pinkish stuff here is now the Enterobacteriaceae coming to the fore, but it's still a long way from being a simple, straightforward genome that's being pulled out of the metagenome. So then he went to the next stage, which was he scratched his head and he thought, how can we get to this? And he thought, was anyone, let's have some audience participation, anyone got a clue as to where you'd go from here? Yeah? We basically said, why don't we subtract everything that's found in normal people? Now, luckily, he didn't have to go out, in the high street in Birmingham and go and collect a load of stool samples from 40 normal Europeans, because they were available already as a project called MetaHit, so you could actually get off the shelf 40, you can get multiple hundreds of different stool samples. He chose 40 age-matched stool samples where the metagenome was known and subtracted from ours all the stuff that was present in the normals. And then he managed, oh, there's the taxonomic classification. I don't know why that disappeared in the end. But in that way, he managed to get us down to the Enterobacteriaceae, down to the outbreak strain E. coli, and he managed to reconstruct the genome of the outbreak strain E. coli purely from the metagenome. So we actually managed to then get our paper published in JAMA, having achieved that particular breakthrough. Since then, I'm coming to a close in the next few minutes. Since then, we've had many metagenomic diversions. We kind of realised that this is a very versatile approach. So I've had a science paper looking at wheat sequences from the Neolithic. We've done metagenomics of medieval skeletons and found Brucella genomes, leprosy genomes. Done a lot of work on the metagenomics of the chicken gut microbiome. And interestingly, even though there are billions of these birds across the planet, and you might think this is a very commonplace kind of place to go looking for bacteria. In fact, half the bacterial genomes we recovered from the microbiome of the chicken by metagenomics turned out to belong to new bacterial species that had not yet been described or named. Now, in the last few minutes, I'm just going to take you on a quick diversion. This is not really to do with E. coli, but I think it's worth mentioning. We probably will do similar things with E. coli in the future, but it just again shows the power of metagenomics. So the story for this diversion begins in a place called Vats in Hungary, where there was a crypt discovered in this church, where there were 242 bodies discovered after a period of over 150 years, undisturbed, and many of them have become naturally mummified for reasons that are not clear. We were interested in this. We particularly said, well, why don't we try using metagenomics on material from these mummies and see if we can get anything out of it. And so we started off with this individual here, Teresia Hausmann, who had died in 1797 at the age of 28. She had a clear chest x-ray. Basically, there had been a previous study done looking for TB in these mummies. The chest x-ray had been clear, but she appeared to be kind of wasted, so consistent with a cachectic, as it's called, consistent with TB. And microbiological and molecular analyses using things like PCR and acid-fast staining had confirmed that there probably was TB present. So we, again, in the spirit of let's just sequence it, just do it. We extracted DNA, sequenced it on the MySeq, which we just got hold of, and we found 8% of the reads that we got out of this lung aligned against TB and tuberculosis. And this ended up providing us with 32-fold coverage of a TB genome from the 18th century. Now, I scratched my head and I thought, well, something must have gone wrong. It can't work this well. But it couldn't think of any other reason why it should be there. We hadn't been growing TB, analysing TB in the lab, and we had to conclude it was real. And in fact, we wrote that up for the New England Journal as a letter, and then we went on and looked at 8 of these individuals from this crypt, and there are their details set out here. And we got variable coverage of TB genomes from these individuals. So you can see in some cases we've got 11 million reads with Teresia Hausmann, 4 million here. Variable coverage. But one of the interesting things we discovered, well, first of all, that they all appear to belong to the same lineage, this particular Euro-American lineage. But we discovered that most of the bodies in our study yielded more than one genotype. And this was rather surprising to me. Sort of second big surprise after discovering that we could actually get TB genome sequences. We've discovered multiple genotypes. And I said, is this a mistake? And then I looked at the literature and realised that maybe it's not a mistake. Actually, mixed infections are still seen quite commonly in high prevalence areas like KwaZulu-Natal. About 1 in 5 patients there, you can detect they've got a mixture. Four genotypes have been reported from a single patient. But the key point is that microbiology, as we do it now, where we pick, like Koch says we should, we go and pick single colonies and then follow them up. You basically obliterate all that mixture that was in the original sample. And it's very onerous to sample multiple colonies. So probably mixed infections are more common than we think. And probably back in the day, this was probably at a time when TB was at its peak in Europe, it was even more common back then. Last few slides, I'm going to run out of time. We basically did some of that kind of genomic astrology known as phylogenetic dating, and came up with a date of about 400 AD for the ancestor of this lineage, which conflicts actually with other studies, which suggests that TB is older than that. And we're currently looking at a Neolithic sample as we speak at the moment, actually. That was interesting, but Bill Gates noticed it, and that gave me bragging rights with the children. But obviously, making a diagnosis 200 years after the event isn't much good. So we actually then turned this on to looking at modern samples, and we did metagenomic detection of TB in sputum. With Emma Doughty, shown here, who did this work in the Gambia, and we're able to actually detect TB in sputum directly with metagenomics, and not just detect it, but in 7 out of 8 of the samples, we could actually assign it to a particular lineage on the TB phylogeny. So that was interesting. So where does this lead us? I'm now just about finished, last 2 or 3 slides now. I mean, one of the directions of travel here is that we could probably do a lot of stuff by sequencing that we currently do using other approaches for. We might converge on something we might call macromolecular monitoring, where we can look at shed DNA, RNA, proteins from urine, feces. We can look at circulating material, taking blood tests. And interestingly, bacteriology and microbiology in general is converging with all these other fields, are also coming to the same conclusion that actually sequencing stuff to find a diagnosis is a very productive way forward, particularly now that sequencing has become so straightforward and you can achieve such depth of coverage. So there's lots of interest in people looking at, say, transplant rejections or fetal health using these so-called liquid biopsies. And it's interesting that the microbiology is moving in the same direction. And obviously with things like nanopore sequencing or any kind of sequencing where you can read a molecule, single molecule at a time, you can perhaps read not just DNA but RNA and proteins as well. Who knows where this is going to lead us to? In terms of my own interests, I'm still keen that we should start liberating E. coli from the lab. So I've presented you some ideas from genomics, but in fact we really have very poor understanding of what makes E. coli tick. And here's a quote from Carl Woese, who made this point, basically, that we have to resynthesize biology, put the organism back into its environment. So we discovered a huge amount from studying E. coli K12, but we have to reconnect this organism now with its history and its ecology. And just recently we've been also seeing if we can do some intelligent design using synthetic biology to improve on nature, to use E. coli as what you might call a smart probiotic, where you could actually get it to do useful things that you program into it rather than just relying on what nature gives you. So what we've been doing is we've been putting in enzymes into the bacterial flagella filament. There's a filament here. We've been putting these onto the surface of the flagella filament in the hope that we can engineer beneficial effects into the E. coli when it's in the gut environment. And we've got this working with just one enzyme at the moment, amylase, but we hope we can extend that. So in conclusion, I've taken you for a romp through E. coli and many other things. We have E. coli here as our mirror on the world, if you like, to get a hat tip to Escher. It's an evolved organism, not a designed one. Still obviously poorly understood, but in a sense it could be our hope for the future if we can redesign this organism. This is a picture also that was in Gödel, Escher and Bach from Magritte, a surrealist painter, and his point here is this is not a pipe. What he was saying is this is not a pipe, this is a picture of a pipe. And what we see of E. coli normally is not a real organism, but some kind of abstraction of what organisms are. Maybe we can move towards a redesigned E. coli. And that's me finished. I just acknowledge all the people that have taken part. And in fact, what I will do is leave the last word, though, to Charles Darwin. He pointed out that there's grandeur in this view of life, with its several powers having been originally evolved into a few forms or into one, and that whilst this planet's gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful, most wonderful, have been and are being evolved. So that's the closing words of the origin of species. It's the only time he ever uses the E word in the whole of the book. That's me finished. Thank you very much.
Thanks very much.